So here we're going to look at, in our course G08 Lab 2 on metagenomics, we're going to look at CheckV to assess metagenome assembled genome mag quality. So if you saw the intro videos, you'll know that CheckV is being designed for viruses, and to a large extent, its estimation error is relatively low in most viruses, with a few exceptions where maybe it doesn't work quite as well. As for Viridae, let's say SFE, ASFE is challenging on a good day. Poxvirus is a bit little bit lower as well. That's understandable. And what it has several processes, and it uses information from different databases to use hidden markup models, HMMs, to iteratively identify what uh, of our contexts are similar or different from the existing contexts in different databases. And these are both virus and host, and therefore it can look at contexts that are either virus or either host, or mosaics of them where we've mixed up the virus and host genes together, which, unless you're talking about retrovirus, shouldn't really happen. And that can be explored looking at gene annotation, gene similarity, and GC content as well. We can also look at take uh, existing contexts and look at the sequence similarity of our context to other contexts to identify what are the most likely contexts to which they match, and therefore estimate the completeness of those genomes that are present. And this uses amino acid quality to compute this, and is able to estimate the difference between the match length and contact length to estimate this. It also gives a confidence score, which is useful, and it also can do this independently without other information to look using hidden markup models that work across the particular contig as well. So in addition, we can look at the direct terminal repeats and inverted terminal repeats, which you can see there in the diagram to predict closed genomes. So you may have genomes that are accidentally placed inside host genomes, which is not desirable, but the direct terminal repeats should be indicators of that. Same with the ITRs, and therefore CheckV will try to predict these as well. So we also get a kind of final summary report on its prediction of closed genomes and the quality of those genomes. Seldom we get ones that are very high quality in reality. Most will be medium or lower. So that kind of is the scope of CheckV to assess the quality of viral contigs, check for contamination, look at completeness, and look at closed genomes as well. To run the full pipeline, it's pretty easy. We use CheckV end to end. We give it the FASTA file as input, and we give it the output folder and the number of threads to use as well. So if we run this, we'll get different tabular data. For the quality summary, this will vary quite a bit. So firstly, we can identify contigs that may not be viral, like contig 1 here. So you can see here it has 11 genes, but at least 2 of them are identified to be host. It's likely that none of them are viral, at least. In contrast, for 2, 3, and 4, we see that many of them are viral, but there may be some host ones as well. And you can see there's some that are quite big with high gene count prediction. There's also a quality score associated with that, and a completeness score as well, based on different models such as average amino acid identity or HMMs for something that's quite diverse. We can look at what output we would get for our viral assembly of the adenovirus. We'd find one big contig, which has 18 viral genes. So there's other unknown genes inside there, and its completeness is very good. It's 98.7%. Whereas the other contigs, they may contain genes, and this one maybe have some viral genes, but it's not likely that they actually have any meaningful information. For the metagenome assembly, what we can see is that this is somewhat different, that there are many host genes in the first contig, and there's some in the other contigs. This contig 3 node 2 2 is quite different in that it has no viral genes nor host genes as well. So those are quite different results. If we look at the contamination, we'll see in this example, contact 2 has a mix of host and virus, which you can see, which you can see there. And then you can also see the contact 4 is also mixed host and virus, which we can get as well. So this is the contamination exploration. For our samples, we see that we have mainly viral here, and some hosts. So it's kind of repeating the same information. So the perspective of contamination depends on what you're looking for. For the completeness, in our example data from the CheckV website, you can see here that we have a certain expected length for the first contig, and its completeness is relatively short compared to the total expected length. Whereas for the second contig, the expected length, the actual length is longer than the expected length, indicating that it's more than complete, and possibly repeats inside there. And if you look at our ones, we can see that the confidence score for the first two are high. Expected length is only a few hundred bases longer for the first contig, and therefore it's very complete, whereas this second one is just a small fragment. If we look at the metagenome assembly, we see that the expected completeness is much longer for many of these contigs, kind of indicating that they're not viral or nor bacterial in origin. Their confidence levels are all low, and the completeness is quite short. So finally, we can look at the complete genome. So in our example data, there's good predictions of direct and inverted terminal repeats for many samples, although the confidence level does vary, and the repeat size does vary as well. If we look at our data, we see that DTRs are predicted for metagenome assemblies, whereas for the viral genomes, that's not the case. The confidence level is not great or not determined in some cases, but you can see that this is the type of output we would get. So let's look at how we do this on the server now. So if we look for CheckV and how to run it, with this structure in our example, where we just have our end-to-end for a particular sample is of interest and our output file. What we can do is use the examples from the worksheet where we're using a higher number of threads just for this tutorial example. We're just going to send it to out virus check2, and we're using out virus context there. So if we press enter, that will now be running. You can see it doesn't give much output to the screen, but that will run away. And I have to check the local database. So I should have mentioned that we need to set our CheckV CheckV database to be this location. So once we do that, and then try our command for the viral context first of all. So this is doing end-to-end pipeline using this as input, sending this to this output. And what you can see there is that it ran very quickly. Firstly, it checks for contamination, then completeness, then it checks for the complete genomes, and then does the quality summary. So we can then repeat that as well for our whole context as well, and then look at the outputs. And these have been sent to different folders. So if we look at the outputs, we can go here and see the viral ones. So you can see there, if we look at the quality summary, first of all, so this is not the easiest to read. So I'll move this over a little bit. You can see there for node 1, we have the length, provirus no, the length, number of genes, viral genes, and its quality level. And you see for the other ones, they're fragments of sequences, but they don't have any real information. If we look then at the contamination, again, we see the same information there for the first contact, which looks pretty good. If we look at the completeness, you can see there that our first contig has this length, this expected length, so it's 98.7% complete with high confidence. Um, if we look at the other fragments, they may contain sequences for some other known contigs, but it's unlikely that they really have any tangible information. And what's the last part then? We have our complete genomes. So you can see that we have none for this. So then if we go back to the sample for CheckV, what we can see there is that we have the same outputs and we can go through them. We can look at the quality scores and we see for the first one is that it's 59 kb, is 55 genes, one of which is viral and 39 host, but it's low quality. And you can see that we have the same results as mentioned before for the other ones. If we look at the contamination, we can see again the same outputs and no real additional information there. If we look then at the completeness, we can see that this one is only a portion of the expected genome length. Is expected to be much longer. And therefore, if the matching metagenome assembled genome is correct, then it's unlikely to directly come from a virus at least, and is very, very unlikely to be bacterial. And then if we look at the complete genomes for this, we see that we have direct terminal repeats for some of these, indicating that they may contain parts of complete genomes, but they may not be fully complete yet, and they're all low complement scores. So you can see that our outputs from the viral genome assembly versus co-assembly are quite different, and we'll explore these in the next video. So please look at that for functional analysis.